Good morning, everybody in Montana. Thanks for joining us today in our Montana State University Montana Agricultural Experiment Station virtual field day question and answer session. We have over 56 videos published on our field day website. Can be found in the news releases and everywhere else. First of all, I'd like to thank Mike Suarez, Sharon Henderson, and Reagan Collier. They really hauled most of the water on putting this all together. And I want to thank all the researchers and people that put the videos together for the producers of Montana. There is some absolutely outstanding videos out there and learning opportunities for the producers of Montana. Pretty exciting for something that was pulled together on a COVID-19 result. We sure wish we were with you personally. We really enjoy field day. At least one of my favorite days of the year is every time I get out with the producers and sit in the wagon and listen to all the researchers throughout the real research centers and other parts of the state, like Bart Farm and Bozeman. First thing this morning, we started out with a soils and soil microbiology and pathology group. So I'd like to introduce you to our three panelists today. Dr. Clayne Jones, Dr. Jed Eberly, and Dr. Frankie Crutcher. At this time, I'm going to give them a couple minutes just to kind of introduce who they are and some real fun facts, and then we'll start right into our questions. Thanks for joining us today, and let's start with Frankie, Eastern Montana.
Yes, so I have been the plant pathologist at Eastern Ag Research Center since 2016. My job here is to come up with strategies to control diseases of important crops here in Eastern Montana. So this pretty much includes sugar beets, pulse crops, and cereals. In my program, we try to really have a well-rounded research approach that includes understanding plant pathogen populations, determining fungicide efficacy, improving pathogen detection, exploring the effect of different agronomic practices, and then really helping the breeders develop resistant varieties. So I try to know a little bit about a lot of different diseases, but I've really had to focus. And so I work mostly with Rhizoctonia root rot, Fusarium yellows, and Cercospora on sugar beets, root rot pulse crops, Ascochyta on chickpea, and then Fusarium head blight has been something I've really focused on. But if you have questions on other crop diseases, that's okay. Please ask them anyways. And if I can't answer, I'll be happy to either recommend someone who might or find that answer out for you.
Wonderful. Thank you, Frankie.
Thanks, Darren.
Let's go to Clayne in Bozeman.
Hi, everyone. I'm MSU's Extension Soil Fertility Specialist. That means I can answer any questions related to fertilizer, nutrient management, keeping nutrients out of our water. My recent research is focused on nitrogen fixation of both peas and lentils, and the video that's shown online talks about an N fixation study I'm doing in collaboration with Peggy Lamb at Northern Ag Research Center, Kevin McPhee, and Perry Miller and Scott Powell down here in Bozeman. Other research that I've been focusing on lately include nitrate leaching in central Montana, as well as the effects of cover crops on nutrient cycling. If you have any questions on any of those or anything related to soil fertility, make sure to give me a call. I also have one other video on the virtual field day webpage, and that's on the growing problem of soil acidification in Montana. So take a look at that if you're interested as well. And I hope to receive some good questions soon.
Outstanding, Clayne. Thank you. Jed, how are things in Moccasin?
Right here, Darren, thanks. My work here at the Central Ag Research Center is focused on soil microbiology, and the role of microorganisms in soil health and crop productivity is really a topic that has been of growing interest and concern in the agricultural community in recent years. You know, people often think of microorganisms as pests that are destructive to their crops and animals as well as to themselves, but many microbes are beneficial. And my work is really focused on trying to understand these beneficial interactions between plants and crops. So microbes are involved in a variety of very important functions, including decomposing plant residues in the soil through the process of mineralization to make nutrients available in this organic matter to subsequent crops. Microorganisms also fix atmospheric nitrogen for legumes. This is probably one of the best examples of beneficial microorganisms in the soil, but there's many other examples as well. One good example of this is the mycorrhizal fungi that can form symbiotic relationships with plant roots and essentially act as an extension of the roots to enable plants to access nutrients that would otherwise be inaccessible. And soil microorganisms play many essential roles in protecting plants from a variety of stressors. This could include things like drought and heat stress, heavy metal toxicity, also protection against pathogens like Frankie was talking about. It turns out that in addition to some microbes being pathogens, there's others that can provide protection against those pathogens. So here at the MSU Central Ag Research Center, my research team is working to better understand some of the key roles that microbes play in enhancing crop health and productivity. So we're working to identify beneficial microorganisms that are locally adapted to our conditions here in Montana. And within this area, we are specifically looking at identifying native nitrogen fixing organisms. And we're looking for organisms that are very efficient at fixing nitrogen, but also robust enough to survive our harsh climate conditions where they're exposed to extreme heat and cold as well as drought conditions. And we're also working at isolating and characterizing novel phosphate solubilizing microorganisms that may be effective at making phosphorus more plant available. So with that, I'll turn it back to you, Darren. Thanks.
Thanks, Jed. Looking right out of the boat, kind of a common question we see a lot of ways is, Frankie, what are we seeing for pathology problems in the state of Montana this year? What should producers be scouting for? And how does the sugar beet crop look in eastern Montana?
All right. Well, I think early in the season, what we saw most was frost damage. We had a lot of episodes of real cold temperatures, especially over here. So I did get questions about that as far as how is this the plant disease or is this something else? Weed streak mosaic virus was an issue this year because last year we had all that rainfall. We had a lot of lodging, especially here in eastern Montana. And so a lot of farmers weren't able to get out and spray and eliminate that green bridge before the fall season. So we did see quite a bit of that as well. Surprise that I saw was powdery mildew on peas. I haven't seen it this severe before, but we're very lucky in that it's occurred near harvest. So if you have seen it in your fields and it's starting to dry down already, you don't need to spray. There's not much you can do about it at this point, and it should have a minimal impact on your yield. And then lastly, what we've seen for sugar beets, we just scouted out Cercospora here on the farm 2 days ago. And so this is about 2 to 3 weeks earlier than we normally see it. And we've got some more humid conditions coming in where you're going to get that water building up on your leaf surface in the morning, which is ideal for Cercospora. So if you're growing sugar beets here in eastern Montana, it's time to go out and start scouting and looking for it. Thank you, Darren.
Thanks, Frankie. So Jed, a question coming in is how do you determine beneficial microbes from just surplus microbes, or how do you relate that back to agricultural production? Kind of a different type of question of more about the philosophy of how you go about that and why there's beneficials or what that constitutes.
Yeah, so one of the challenges, well, I guess back up, we've known there's beneficial microbes. That's been common knowledge for many years, especially within the research community. But really the challenge has been trying to identify them and determine how many we have present in the soil and what functions are they performing there. So that's been challenging. In recent years, there have been a number of measures of soil health that have included aspects of analyzing the microbial community. So a few that have become, or probably the best known, are the Haney Test and also the Cornell Comprehensive Assessment of Soil Health, or CASH. So these tests measure soil respiration, and they can provide an indication of things like microbial biomass in the soil and also the potential activity of those organisms in the soil. So some of them can also incorporate measures of potentially mineralizable nitrogen, which again, that's another way of getting some insight into microbial activity. And then there's also more direct measures of root pathogens that can be performed. And that's looking more at the part of the community that would be detrimental to the crop. So the downside to some of these approaches, though, is that they really don't enable us to directly quantify the beneficial microbes in the soil. So this is still a big challenge for those who are wanting to take a more holistic management practice that incorporates an understanding of the microbes and using agricultural practices that are of benefit to the microbes to help enhance their activity and performance in the soil. There are numerous new technologies that are under development. A lot of these are sequencing based. And while in the past they've really been too expensive for application by growers, they've been pretty much restricted to researchers. In recent years, as the technologies continue to develop, costs continue to decrease, there are some labs starting to offer this as a service to growers if they want to get a more comprehensive understanding of the beneficial microbes in their soils. And there are, some of the more traditional methods of doing direct microscopy to try to identify organisms. But again, some of those approaches are more expensive and time consuming. But I really think we're on the verge here of seeing some rapid developments as economies of scale kick in and these tests become more routine. They'll be applied at a much broader scale for growers to gain some of that information. Back to you, Darren.
Thanks, Jed. Hey, Clayton, it kind of came in from your perspective as a soil fertility specialist. How does that interact your position and what you do for producers and Jed's work with the microbiology or microbiome? How does that interact with how you two see that?
Yeah, that's a good question. I think, Jed is getting a lot more into the soil biology. I would say that I address more the soil chemistry, realizing that the two are not separable. So I understand that what drives nutrient availability, so available nitrate, phosphorus, potassium, for example, is largely microbially driven. So I have to understand a little bit of what Jed does, and Jed certainly understands my side of the business. I say I would probably use more traditional tests, laboratory tests that we've calibrated over decades in Montana to reflect nutrient availability, but we recognize that those are done in the lab and don't always reflect nutrient availability in the field. And that's what some of these newer tests and what I think Jed's trying to get after.
And Darren, if I could follow up on that.
Please.
As Clayton mentioned, it's this intersection between the chemistry and the biology, and also the physical properties to some extent play in there. So really from a research perspective, what we're looking for is what are the best metrics that are easy to measure and economical that we can use to give us an understanding. So that could potentially include chemistry measurements that provide insight into what's happening in the microbiology, or it could be some microbe-focused measurement that would provide us some insight to what's going on in the chemistry. So these very much play an interactive role together.
Perfect. Thanks, guys. Clayton, not surprisingly, from the Triangle area, top 5 things we're working on, and how does acidic soils impact? We know it's a big problem in the Triangle right now, and you're working on that. So could you address some of the acidic soils and maybe a five or so recommendations, or what you're thinking for recommendations on acidic soils?
Sure. So a little background in case you haven't heard much about this growing issue. In about 2015, we discovered some acidic soils on the Highwood Bench in Choteau County, which was a surprise to me and other soil scientists because Montana's generally had alkaline or high pH soils. Once we discovered it and started looking for it, and crop advisors, producers, extension agents started looking for it, we have now found this problem of acidic soils, meaning low pH soils, in 24 of our 56 counties. Choteau County is still the epicenter, but we're finding it in additional fields. Things to look for are stunted growth, yellow to pink to purple, lower leaves. We also are seeing more herbicide carryover. So if you're suddenly starting to see more residual herbicides in pockets on your field, that can indicate low pH. Poor nitrogen fixation or nodulation of legumes is another good indicator. In fact, we call it an early indicator because the rhizobia that make the nodules and fix the nitrogen on plant roots actually start showing problems around pH 5.8 or so, where we don't see real detriment to crops till about pH 5. So it's kind of an early warning system. I like to think about mitigation, adaptation, and prevention. So mitigation would be primarily lime, often applied at high rates, 1 to maybe 3 tons per acre. Adaptation would be selecting crops and varieties that are more resistant or tolerant to low pH. Winter wheat seems pretty tolerant. On the other end of the spectrum, safflower and the legumes and durum wheat are very intolerant. And then also phosphorus can be applied with seed to tie up aluminum, which is the problem caused by low pH. And then as far as prevention, anything you can do to reduce nitrogen rates. So that would include things like using more legumes in rotation, planting crops that need less nitrogen, such as perennials and barley, and making sure that you soil test for nitrogen so that you don't over apply nitrogen. So in a nutshell, those are some of the things you should be looking for and some of the management practices you can do to prevent or correct this problem.
Thank you, Clayton.
Clayton, a follow-up question on that. You mentioned phosphorus. Have you looked at all at the economics of that? I mean, understanding that that's probably more of a temporary solution, but compared to liming, how does the economics of that play out?
Yeah, so in a study done by my colleague, Rick Engel, he found on one site in the Golden Triangle that phosphorus doubled durum yield from about 22 bushels per acre to about 43 bushels per acre. We were applying 60 pounds of P2O5 per acre, which is pretty high rate, but you do the math, that would easily pay for itself. But in the long term, phosphorus is not fixing the problem. It's kind of helping in that particular year. And so my back of the envelope calculations show that in the long term, lime is a much less expensive option and truly fixes the problem for an extended period of time, meaning 1 to 2 decades.
Thanks, Jed. Good question. Thanks, Clayton. Frankie, I think people are picking up on a theme this morning, but could you talk to me on how these soil-borne diseases may creep up on people or kind of build on themselves?
Yeah, and that's definitely something we're seeing, especially with pulse crops. When in the scheme of things, pulse crops are still relatively new to our cropping systems here in Montana. There's 2 really big reasons why we're starting to see these proliferate now. One of them is because just presence of the host, and especially since a lot of these pulse crops especially are being grown in really tight rotations. So the more years that you have the host plant, the more that the pathogen is able to proliferate, make spores that can stay in the soil for a long time. So a really good example of this is Aphanomyces on pea and lentil. This is something that's becoming an increasing problem, especially here in eastern Montana. It's very host specific. It makes spores that can stay in the soil for 10 to 20 years. And it takes time for it to build up. And so what we've seen and what producers have noted is that it takes 5 to 6 growing seasons with pea and lentil for you to start seeing symptoms. So this is why it's really important to diversify rotations, to lengthen the time that peas or lentils are in your field when it comes to Aphanomyces or other root rot pathogens that we're seeing. It's also important to know what you're working with. If you see that plant that's not looking so good in your field, it's good to pull it up and look at the roots. A lot of times people forget that these diseases can be on the roots. Send it into the Scudder lab. They have a quick turnaround time, and they can give you some really good information so you can start making decisions on if you want to change up your agronomic practices or your rotation schemes, if you want to add a more specific seed treatment, or if you maybe want to choose a resistant variety, try something a little bit different. And so knowing what you're dealing with gives you more choices. So,
good.
the second reason actually has to do with Jed. Having diverse microbial life in the soil limits pathogen activity. And so knowing more about that is obviously very important for controlling plant pathogens.
Right. So really quickly, Frankie and Clayne, you both talked a little bit about the past two topics about variety or resistant varieties. And I know we're going to have our breeders on today, but in both of your cases, maybe acidic soils and in some of the disease prevention, how do you guys interact with these, and what do you see on the forefront in the breeding programs?
Why don't you go first, Frankie?
Okay. I'm really blessed to be working with a group of breeders who take plant diseases very seriously and are very active in breeding for those. So for our cereal breeders, they're all very focused on Fusarium head blight and have put a lot of effort into starting to breed for this resistance and get out some good regionally adapted varieties for us. I have a nursery here in Sydney, and we have one for winter wheat down in Huntley, where we're screening these different progeny and varieties that are recently are going to be or have been released, so that you can get really good up-to-date information on that resistance. For the Pulse program, Kevin McPhee has focused on a variety of diseases. Him and I have started a project to start breeding for better resistance to Ascochyta for chickpea, which really plagues producers who want to grow chickpeas. It definitely has to be one of the big determining factors on whether or not they're going to grow it. Because once you've had a couple seasons of it, it really starts to become a problem. If the weather's right, you could be spraying 4 or 5 times. So adding a resistant variety would be beneficial.
On the acid side of things, all 5 of the breeders, so Jamie, Phil, Luther / Jason, Kevin, and Mike Drew have all been working on tolerance to low pH in acid soils, which is pretty impressive because it's still in some ways a relatively small problem in Montana. It's a big problem for certain producers though. Rick Engel, Pat Carr, Simon Fordyce have all been working up on the Highwood bench, sometimes in collaboration with these breeders, looking for varieties that are more tolerant to low pH and what the end result of low pH is, which is high aluminum in the soil. The research is extremely challenging because none of these sites are uniformly low pH. So we end up with high variable results. And like with any breeding program, it's going to take time to develop varieties. Fortunately, some of our colleagues over at Washington State have developed some spring wheat varieties that are tolerant, and those are actually right now being grown in Montana. We also have discovered varieties that appear to have aluminum tolerance, even though it wasn't bred for aluminum tolerance within Montana. You can find a list of those varieties on my webpage. And if you want to find my webpage, just Google Clayne, C-L-A-I-N, and soil, and it's usually the first hit. Then look at the soil acidification page on the left-hand side.
Wonderful. Thank you, guys. And so if there's more questions for the people in the public, we will have the breeders on. They'll be on between 2 and 3 this afternoon if you want to get into specific varieties and some of those different recommendations. So thanks for bringing that attention. And I'd be remiss if I didn't say that all the researchers you see on your screen today drive huge collaborative work. And that's just one example of what you're seeing here is between breeders, between pathologists, between soil scientists, microbiologists. We all push on the same side of the rock and try to move Montana agriculture forward. And that's a pretty proud thing for me to be able to say because it is a team effort at Montana State University and we're here for the producers. So Jed, we're going to take a little bit of a step back, and a producer from Liberty County is asking that in recent years, most of the hype about soil health has been with cover crops, and how the cover crops impact soil health, and how do they fit as a replacement in fallow when we don't get more than 7 inches of rainfall in our growing season. Could you talk a little bit about your work you've done with cover crop and how you see that impacting it, please?
Yes, that's a really good question, and it's one that's very relevant to producers around the state. I can't speak so much to the water use component or the yield component, either of the cover crop or subsequent wheat yields, but speaking specifically to interactions within the microbial community, the results really are quite variable. We do see some evidence that by increasing diversity through incorporating a cover crop, you have greater species diversity in your crops that you're growing. That can have a benefit to the soil microorganisms, but it's not consistent across all locations, regions, or cover crops. In general, though, it does seem to be a good thing. We are seeing in some of the work up at Northern Ag Research Center that incorporating some warm season crops does seem to have a benefit in increasing the microbial diversity, and specifically increasing a few of the beneficial microbes that are present. Now, one of the things we're still trying to sort out is this benefit due to the fact that we have warm season cover crops in the mix, or is it simply more of a timing issue of when those crops or those cover crops are being planted? So when you're looking at some of our cool seasons versus the warm seasons, you can have a fairly big time difference there between when they go into the ground. So that really impacts the amount of root biomass that's present later in the season, given the available moisture. You know, if you're planting later and you don't have sufficient moisture, you could get less biomass in the soil there. So there's a lot of variables that play into this. In general, though, I would say the results are positive in terms of benefiting the soil health, but there's a lot of subtleties that go into that we're still trying to better understand.
Thanks, Jed. Clayton, you work a little bit with cover crops. Do you have anything to add to that kind of thought process?
Sure. I mean, it's a good question. Can cover crops really make it here in Montana, where we have low precipitation? And certainly economically, work done by my colleague Perry Miller would suggest when we spray out cover crops in a no-till situation, it's really hard to make them pay because of the cost of the seed and the management, and relatively small benefits to the subsequent crop, and often a detriment to the subsequent crop because of water use. Where we think cover crops might have a fit is more as an annual forage. Work done by Darren and others up at Northern Ag Research Center have looked at the effects of grazing and haying versus spraying out. And at least preliminarily, it doesn't look like removing that biomass has a really, that above ground biomass has a really detrimental effect on soil health, which is a good sign, because certainly haying and grazing have an economic benefit.
Yeah, that's a good point, Clayton. We will have livestock interactions later on today. I believe that's between 1 and 2. And although we're doing a full economic analysis with George Haynes and Anton Beckerman of an 8-year cover crop wheat rotation trial, it looks from just cash receipts that adding livestock, specifically beef cattle, to our grazing, as a grazing a cover crop was able to compensate for the wheat drag the following year with the water use, to get back to the question or the comment you had, Jed. So we can sure talk about that a little more later on. But definitely, I think integration of livestock or hay an option to offset that until we can build that, you know. And can you talk about how that change occurs, Clayton? You know, we have a pretty good documented track record in Montana of, you know, increasing organic matter when we went from cultivation to no-till. So how do you see it as a soil scientist, us changing as we move into maybe this alternative cropping systems or cover cropping, and how long or what we can predict maybe on how we have to get on the train and kind of follow a plan to change organic matter or to change our soil profiles? Maybe Jed could jump in on that one also.
Yeah, take home message is in a low rainfall area like Montana, it's very hard to change soil organic matter. And the reason for that is we just don't return a lot of biomass to the soil. Preliminary work by myself, Perry, Rick Engel, has estimated that maybe 40% of the biomass that's returned ends up as soil organic matter. But when you start with something on the order of, say, 15 tons of soil organic matter per acre in the soil, and you're returning on the order of maybe 1 ton per acre, and then 60% of that actually ends up decomposing and going to the air as carbon dioxide, you can envision it takes a long time. We are able to detect change in our long-term trials where we convert from fallow to recrop or perennial or cover crop systems, but usually only after about 6 to 8 years can we really detect change. So the take-home message is it took a long time to decrease soil organic matter. It's going to take a long time to increase it, but if you're patient and you look decades to centuries down the road, it's certainly doable.
Great.
And Darren, if I could follow up on what Clayton mentioned about termination, one of the things that we've found in this study is that there really seems to be no difference in the microbial community profile if you terminate by chemical methods versus grazing or removing it as hay. So I think that's really a positive thing in the context of making this profitable for the growers, that if they are wanting to harvest it for feed, it's really not going to have a negative impact relative to chemical termination.
Great.
If I could comment from a
Please.
pathology perspective. So I get this question a lot for people who want to do cover crops, and that's if they have peas in their cover crop mix, are they going to build up inoculum to make their root rots worse than their pulses in the future? And that's a question that we're definitely looking at right now. I'm working with Audrey Carlisle over at NDSU. We have 1 year of data looking at root rot in cover crop systems of peas, and we found that it was significantly lower than in our monocropping systems. So I'm hoping to get some more years of data out of that so we can really answer that question for producers, because that's the last thing we want to do is make it more difficult to grow the actual crop.
So Frankie, that brings up a question I get quite often, and maybe you could answer this from a pathology standpoint.
Sure.
The rotational requirements or the rotational recommendations between peas and lentils and insurance reasons kind of takes cover crops out of it if there's any kind of pulse pea or legume in there, right? So how do we talk about that, and how do we think about that? And the work you're doing, is there any other historical work you know of to help us make that decision on how to do that? Because they kind of want the legumes in those cover crops.
Right. Not that I'm aware of. I'm sure they're doing work out of Canada. They have very much more strict recommendations for rotations in Canada, 5 to 7 years between lentils and peas. Lentils and peas being the same under the soil. Microbes see them the same, so it's pretty much the same conditions. Here we recommend 3 years. But like I said, the answering that cover crop question is really important. Though I have not heard is it considered that for insurance purposes? So I think is where the question really comes out. Yeah.
Being able to, you have to be a certain number of years, and I've forgotten how many years between the cover crop, even if it has peas in it, and that. So that's important data I think we need, not just as producers, but for an industry as a whole, including the insurance. So thank you. Good point. Yep. So Clayton, this brings up another question I hear from Plentywood, or we're getting from Plentywood, is what can we expect if we're returning this biomass to the soil, whether it be a pea crop or whatever, on beneficial nutrients to the next crop, and how can we see that happening, or what's the speed of that?
The beneficial nutrients can be a faster response, especially if there are legumes either in the crop or the cover crop. We think that legumes pulses fix on the order of about 100 pounds of nitrogen per acre per year, depending on moisture. We can be much higher than that and somewhat lower than that. Most of that nitrogen leaves in the form of grain. peas and lentils. Chickpeas are very good at getting the nitrogen into the grain to help the next crop. And that means we might be leaving behind something on the order of 40 pounds of nitrogen per acre from a grain crop. Of that, a lot becomes soil organic matter. So that's why we only credit following pulse crops somewhere around 10 pounds of nitrogen per acre. Following a cover crop might be, that's heavy on legumes, might be more like 30 or 40 pounds of nitrogen per acre. So there can be more immediate benefits nutrient-wise than the long-term buildup of soil organic matter. Did that answer your question?
Yeah, I think so. I'm sure the person will redirect if we need it. And I think my interpretation is how to get that nitrogen credit from growing the pulses the previous time. So I think you did good. So Jed, a question coming in. I didn't say where this location was, but you mentioned the Haney test and the cash test and some of this. So could you talk to a little bit about how producers should be going about testing and some of the soil microbiology work and how you would recommend them doing those tests, or if they want to get involved with that, or when they should?
Yeah, so a lot of the soil testing labs are starting to offer some of these additional soil health tests. They're being promoted from different sources. Basically, it'll require some additional sampling or slightly different conditions when you're doing those sample collections, depending on the test that's being run. But with a lot of these anymore, you can submit it to a service lab, and they're going to provide the results back for you if you're asking for something like the Haney test or the cash test. Getting into the specifics of what some of those tests do, a typical component is looking at respiration rate. So for those tests, they're basically taking a sample of soil, they're wetting it to stimulate the microbial activity, and then you get this burst of carbon dioxide that's produced, and they measure that. And there's a relationship between that carbon dioxide that's produced and the number of microbes, of active microbes that are present in the soil. So there's others too that look at things like the potentially mineralizable nitrogen, also looking at the carbon and nitrogen fractions that are associated with the microbial biomass in the soil. And often with those results, they will give a recommendation or somewhat of an interpretation of what this means in the context of the health of your soil. I feel like this is an area that is really still growing. It's quite new. A lot of growers in our region are probably not familiar with these tests. I am aware of some that are using them routinely. But again, it's what I see this as being another piece of the puzzle. And the more pieces we can add to the puzzle, the more complete the picture that we're able to see of our fields. And again, there's a lot of variables that go into this. Just to build a little bit off of what Clayton was talking about, because I think this ties right in, wondering how much nitrogen credit you're going to get back from your cover crop. Well, there's a microbial component to that. If you have a soil that's very degraded and low levels of microbial activity, it's likely going to take longer for that nitrogen to be turned over and made available to the subsequent crop. Where if you have a field that's been under practices that have really promoted soil health and it has a very abundant and active microbial population, some of those nutrients might be turned over much more rapidly. So some of these soil health tests are really attempts to try to help us understand what that potential is there. And as we look at things like potentially mineralizable nitrogen, and then also with a CO2 burst test, getting an idea of the microbial biomass, we can start putting some of these factors together to give us a better understanding of the health of our soil, but more specifically, how things like nitrogen will be cycled and made available to our subsequent crop.
Great. Thank you, Jed. Frankie, in from Daniels County, can you predict, and you might have to do some preliminary work in explaining this a little bit, but can you predict or have an estimate of what the vomitoxin levels may be come harvest time as we're moving through this year? And maybe you should explain what vomitoxin is.
Okay. Vomitoxin, which us researchers commonly call Dawn, so it's the same thing, is a mycotoxin made by Fusarium headblight in that disease. It's not cancerous or anything like that, but it does cause feed refusal in certain animals. And it can make people sick. So there are limits when you take your grain to the elevator on how much vomitoxin that you can have. Unfortunately, I cannot predict how much we're going to have because as with most plant diseases, it's very weather dependent. If you are in an area and you have it, you went out and you saw it and you know that you have it out there. If we continue to have high humidity, if you're continuing to irrigate, then it's going to get higher as the season progresses. So knowing that you have it is the first thing, and then you can start making decisions and you can start deciding how much you're going to clean it. Because if you know you have head blight, highly recommend cleaning your grain thoroughly before you take it to the elevator.
Thank you, Frankie.
Sure.
I'm going to jump in with a question myself because each one of you today has spoken a little bit about different management techniques for what I would call like an integrated management things for each one of your disciplines. Maybe if each one of you could address some of the things that your recommendations to help the pathology, Clayne, maybe on the nutrient profile and some of the things that you work on in your program. I think I'm seeing a trend of diversification, integration of other alternative methods of farming, cultural practices. If you don't mind, could each one of you kind of maybe address some of the things you should be talking to producers about and maybe should be considering and integrating in their system? And let's start with Jed. You look a little lonely there, still in moccasin.
All right. Well, one of the things I would emphasize is soil testing. I know it's expensive and it takes time and it can be hard to fit it into a busy farm schedule, but it's really essential to know what's going on. I was speaking with a grower here just this week who had done some sampling to look at the microbiology within his field and was surprised to see that in a particular area, there was quite a bit of variability in the pH, one area being quite a bit lower. So I encourage you to go back and follow that with some more thorough sampling across his field. And rather than just looking at pH, let's see what some of the other nutrients are. And he also discovered in doing that he had a lot of variability in his nitrogen. So I've also been working with him to do some of the microbial community analysis as another piece of that puzzle to try to figure out what's going on in different parts of his field. So I just use that as an example. You go out and pull a few soil samples from your field, gives you a little bit of a picture, but as Clayne mentioned, specifically with the acidity, it can really vary across the field. That can also occur with a lot of these other parameters. So just the importance of sampling. And also too is moving forward as we're more aware and focused more on sampling related to soil health, think about the questions that you're trying to answer with the sampling. It's not really sufficient just to say, okay, I'm going to go out and sample everything and do every possible test under the sun. But in the context of your management practices and what you're hoping to achieve with your land, try to tailor your So I guess the one thing I would really focus on in this holistic approach is sampling and the right type of sampling.
So you're really talking about precision agricultural management of specific soil testing is that kind of what you're thinking, Jed?
In a sense, yeah, it really does play into that.
Great, thank you. Clayne.
Yeah, to follow up on what Jed was saying, I know sometimes he did mention the cost of soil testing. I see soil tests somewhere on the order of about $40 per test. Often that's done per about 100-acre field. You do the math, that works out to be 40 cents per acre. Often we find nitrogen levels that might be 10 to 30 pounds of N per acre different year per year. You run the math on over or under fertilizing by 10 to 30 pounds of nitrogen per acre per year, and that soil test easily pays for itself. So as far as integration, you probably heard a lot of people talk about the 4 R's, meaning you have to apply the right rate, source, placement, and timing. If you focus on just one of those, say rate, and don't think about where you apply the nitrogen or the phosphorus, the potassium, you might be wasting some of your fertilizer dollars. I also like to add a 5th R, which would be the right rotation. And so as we've all been talking about, rotation really plays heavily into good fertilizer management. By using a range of crops, we end up helping with disease. By using legumes, we end up reducing nitrogen needs. And so it's really important to think about those 5 in concert. I think in Montana, with a lot of hard red wheat being grown, we probably focus on nitrogen a little too much, and I'm partly to blame for that. I talk about nitrogen a lot, but what I'm seeing is a lot of producers who say, I'm not getting the protein I used to get. Why? And when I look at their tissue tests or their soil tests, a lot of times another nutrient is low. For example, sulfur is also needed to make protein. So really looking at all the macronutrients and the micronutrients, where they're applied, how they're applied in concert with rotation, I think is really key for good nutrient management.
Outstanding. Thank you, Clayne. Frankie.
Okay. Well, I would say, Jed and Clayne and I probably agree on just about everything except for one topic, and that's tillage. So, no-till systems have been great for improving soil health and preventing root rots, and that microbial diversity is incredibly important. But if you're having problems with a residue-borne disease, then dealing with that residue becomes very important. So if you're you're growing cereals and you're having problems with tan spot or head blight, or if you're growing chickpeas and you're having problems with ascochyta, then minimizing your residue on the soil surface becomes incredibly important. And that's the time to really look at and see if maybe a light disking or breaking up that material a bit would be beneficial to you. So I don't ever recommend a hard and fast no-till system. It's look at the situation you're in, look at the problems you're dealing with, whether it's plant pathogens or weed issues, and make a decision based on that as well.
Outstanding. We just got a couple minutes left, and I want to thank you all for this, but I have one more question that I guess that's pointed at me, so I apologize to our questions here. Is Bob from Conrad asks that there's been several recent departures in the research centers. What's the current status and what does it look like in the future? And Bob, I want to say thank you for the question. I feel bad taking away from our panelists here today, but it's very important. The research centers have just recently hired 3 new faculty members. This is in some very difficult times with the pandemic. The Board of Regents did put a hiring freeze on all positions throughout the university process. However, we were able to petition through my office, through Dr. Bajwa's office, up through the provost and the Board of Regents, and they supported 3 new hires out in the fields where there's boots on the ground for research. We have a new weed scientist starting at Southern Ag Research Center, Dr. Livrit Shergill. He'll be taking over for Prashant Jha. He'll be working on specifically in herbicide resistance, crop rotations, and herbicides as a weed scientist. At Conrad Station, we just hired Dr. Justin Vetch. He'll be taking over both a new faculty position and the superintendent position in Conrad at Western Triangle. That's an outstanding hire for us. And over in Northwestern Ag Research Center, we just hired Clint Byerman, who is going to be a cropping systems agronomist. However, he has a weeds background and is very interested in them, or maybe those smaller acres, high dollar return on a high moisture regime in Northwestern Ag Research Center. So I appreciate the question, Bob. I appreciate everybody keeping us on task and making sure our staffing at the research centers, and I would include all of maize, is very important to the producers of Montana. So that's our current status. If you have other questions, feel free to contact me. My email is dboss@montana.edu. My door's always open. I'm happy to field any other questions any other time. I'd like to go around the room, and if you don't mind, Frankie, how can people get a hold of you?
Well, I'm over here at the Eastern Ag Research Center, so you can email me at frankie.crutcher@montana.edu.
Thanks, Frankie. Jed?
Yes, and I'm here at the Central Ag Research Center up by Moccasin, and you can reach me at jed.eberly@montana.edu.
Clain?
You can reach me at clainj, CLAINJ, @montana.edu, or call me at 994-6076 with any soil fertility question.
Boy, thanks so much today for being with us this past hour. It seems like it was 5 minutes. I don't know about your end, but it seems like we did really good. Some outstanding questions. I will follow up with the three of you if we have any questions come in that may pertain to you or interact with the breeders. And with that, thanks so much and have a great day.
Thank you.
Thank you.